The overall goal of this procedure is to produce a genetic cross of the rodent malaria parasite Plasmodium yoelii in order to generate recombinant progeny. This is achieved by first injecting a group of mice with a mixed inoculum containing the two parental clones of the parasites to be crossed. Infected mice are then used to feed mosquitoes, inside which the gametes of each parental strains will fuse into a diploid zygote. Meiotic division at this stage can lead to genetic recombination, thereby producing recombinant progeny. The resulting sporozoites are then harvested from the mosquito's salivary glands and re-injected into naive recipient mice in order to obtain blood-stage parasites, among which are individual recombinant clones that can be isolated by limited dilution. In a successful experiment, 5 to 10% of the cloned lines will be recombinants, which can be characterized by genotyping for appropriate genetic markers.
This method can help answer key questions in the malaria research field, such as what are the genetic determinants of drug resistance, virulence, and transmissibility in malaria parasites.
At room temperature, thaw 2 vials containing the frozen blood-stage malaria parasites to be crossed. In this example, the 2 rodent malaria parasite strains used are Plasmodium yoelii and Plasmodium yoelii nigeriensis. Fit a syringe with a 22